The overall goal of this procedure is to deliver robust and repeatable bacterial infections to Drosophila melanogaster, then to subsequently measure infection severity and quantify the host immune response. This is accomplished by first delivering the bacterial infection into the flies using a nanoinjector. Host mortality can be tracked post-infection over time, and the systemic bacterial load can be determined at any point by homogenizing infected flies and measuring the number of bacteria present. The host immune defense can also be quantified by measuring the expression level of antibacterial peptide genes. Ultimately, quantification and characterization of bacterial infection in Drosophila melanogaster can be used to test how genetic and environmental factors alter immune competence of the fly.
This method can be used to address key questions in the field of insect immunity, such as how host genetic and environmental factors influence immune capability, or how distinct bacteria vary in their infection kinetics.
Although we are using this method to measure immune defense of Drosophila melanogaster against bacterial infection, it can also be applied to other insects and can make use of other types of pathogens like viruses or fungi.
A common problem people experience when they are first learning this method is that they may cause too much injury to the flies, which can result in excessively high mortality even in controls.
Visual demonstration of this method is critical for learning how to do infection with minimum damage to the fly. Since our host species, Drosophila melanogaster, is so small, infection must be done under the microscope, which adds to the initial complexity of the technique. However, we believe that a small demonstration of the technique will render it accessible even to scientists who have only limited experience with flies.